Hello, my name is Alex Book, and today we'll be presenting the effect of cinnamon and peppermint on the inhibition of E. coli and Staph aureus. Peppermint dates back to 1500 BC, where it was first used by the ancient people for food and herbal remedies. Peppermint oils are made up of EO, derived from the leaves of M. pepperdine. Peppermint oil exhibits antimicrobial, antiviral, and antifungal activities against various bacteria and yeast. Peppermint oil uses terpene ingredients effectively to kill bacteria. Cinnamon oils. Cinnamon oils are from dried bark of trees of cinnamon. Cinnamon oil was first used by the Romans and Egyptians for medical properties in the digestive system, as well as preservation of mummies. Cinnamon fights bacteria in many ways. Components include antibacterial activity against gram-positive and gram-negative bacteria. E. coli. E. coli was first discovered by Theodore S. Rich in 1885. E. coli strands colonized from the gastral tract of intestines of humans and animals. E. coli is most common bacteria for its simplicity, well-known genetics, and many molecular tools to react with it. For example, essential oils are used to create antibiotics. The oils reverse the resistance of the E. coli bacteria to the antibiotic. Introduction of Staph aureus. Staph aureus was first discovered by Sir Alexander Ogston. It is a gram-positive bacteria and is in a wide variety of clinical diseases. Staph aureus is found in skin and soft tissue infections, for example, as boils and other skin diseases. Staph aureus is often treated with essential oils like tea tree, which is proven to reverse the effects of the skin disease or other Staph aureus examples. Introduction of Staphylococcus aureus and E. coli. Out of the reported outbreaks in the United States from the time period of 93 to 97, bacteria was the root of 86% of cases. The variety of the bacteria made them of vital importance. The organism's spore-forming capabilities and toxic production made them very harmful to humans. Gram-negative bacteria had a slightly more resistance quality to the essential oils when compared to gram-positive because it was present in many other membranes. I'd like to introduce my colleague, Holden.
Thank you, Alex. My name is Holden Westman, and so here we just had a chart kind of going into further some of the adverse effects of having like food poisoning and how Staphylococcus and E. coli both contribute to food poisoning. So a little bit more into the introduction for essential oils, both of them together. Not much research has gotten documented for these essential oils. The efficacy of thyme, oregano, and savory weren't even documented until the 1950s, and large differences have been observed due to the different harvesting techniques of different time, geographical region, and so forth. But it has been proven for thyme oil to have full antibacterial properties, so it made sense for us to try to experiment with cinnamon and peppermint oils. So a little bit more, Turkish oregano, among other essential oils, have strong antibacterial properties against foodborne pathogens such as Salmonella. So that again kind of relates to why we would maybe try the resistance to E. coli and Staph aureus. And oregano essential oils have also been proven to enforce antibacterial activity. So the reason we had this study was to investigate the potential antibacterial activity of cinnamon peppermint oils, and again, both of those on E. coli and Staph aureus separately. And we also wanted to see if different essential oils provide any antibacterial activity on E. coli or Staph aureus, and then a significant difference would be demonstrated between the different types. So in our procedure, we used nutrient agar plates, which were inoculated with the E. coli and Staph aureus. Cinnamon oil and peppermint oil were then obtained and inoculated onto paper discs. They were placed on each section of the plates, and they were incubated for 48 hours at 37 degrees Celsius. After, I believe, 1 week, we measured in millimeters the zone of inhibition, and we used Excel for our statistical analysis. So here's our first chart here for E. coli. We found E. coli was statistically different for only the peppermint oil, as well as the difference between peppermint oil and the cinnamon oil.
I'm Aaron Feiner, and I'll be wrapping up the rest of this presentation. So looking at our next graph, just showing the inhibition of cinnamon and peppermint, this time on E. coli rather than Staph aureus. We see that the statistical significance is only found in the cinnamon oil and not in the peppermint oil, and once again just showing the control for reference. So our findings just prove that our prediction was correct, that there was a difference in the oils in the cinnamon versus peppermint in terms of Staph aureus and E. coli. The inhibitory ability of both cinnamon and peppermint were shown in Staph aureus and E. coli respectively. And our results, once again, showed that peppermint and cinnamon oils were effective in the inhibition of E. coli and Staph aureus, respectively. Cinnamon oils were good inhibitors just because of the compounds which is in it, which in our research was cinnamaldehyde. It's good at destroying the cellular membranes of bacteria, which lyses the cells, which ends up killing them. As well as peppermint oil ends up lysing cells as well, with a different compound, in this case terpene. And both oils were able to lyse the cells using different compounds and on different cellular membranes between the two bacterias. Our findings demonstrate the efficacy of peppermint and cinnamon essential oils in the inhibition of the growth of two certain types of bacteria, specifically pertaining to the compounds within the certain types of oils and the compounds within it. Our data could help improve the efficacy of bacterial drugs, and not the replacement necessarily of the pharmaceuticals within it, but just the addition of our compounds found within cinnamon and peppermint. Once again, through our study, we were able to find that peppermint and cinnamon oils were able to inhibit the growth of Staphylococcus aureus and E. coli, respectively. So some of the limitations of our research, 3 of them were that we only used 2 types of bacteria, although one was gram-positive and one was gram-negative. We don't know completely how they would work on other types of bacteria. We also only used it at 1 temperature. A different type of temperature could deconstruct our compounds, which we found within the oils. And then we also only used 1 brand of oil, so we don't know how it was collected or when it was collected, and whether that affects the efficacy of our inhibition. So here are Some of our works cited. Some more. Some more. Thank you.